Hi everyone, it's Mrs. Williard and you're watching Enzymes for the unit Ecology. And enzymes are really important. We've been talking about two processes lately, photosynthesis and respiration. And enzymes are partly responsible for how these things can happen so quickly. But there's a lot of things that are going in for photosynthesis and respiration and a lot of things coming out, but it happens really, really quickly. We know we breathe in oxygen and we exhale carbon dioxide really quickly within a matter of seconds. So enzymes are what help that process happen quickly. So there's 4 things you need to know about enzymes. First one is enzymes are proteins. So enzymes are a type of protein molecule. Okay? They're made of amino acids. Every molecule, so proteins, lipids, nucleic acids, those types of things, carbohydrates, all have building blocks, which are called monomers. And proteins is amino acids. And we'll talk a lot more in detail about proteins and how they're actually made later on in the semester. But right now, you just need to know that they are proteins and they're made of amino acids. Another thing you need to know about enzymes is that they have a really specific shape. All proteins have a shape that's very, very unique to them. Enzymes are no exception. And we'll talk about in just a second what happens when that shape gets messed up, when that shape's disrupted, what that does to the function of the enzyme. You can think a lot about it like a key. If you have a car key and something happens to it and it gets bent or whatnot and you can't bend it back in place so easily, it's no longer going to open your car door. So enzyme shape gets messed up, it's no longer going to be able to perform its function. Number 2 is enzymes are catalysts. Catalyst and enzyme, those words are interchangeable. You may have never heard of the word catalyst before, but what it means is that it's something that speeds up reactions. So chemical reactions such as photosynthesis, such as respiration, which respiration in us also involves digestion. It's speeding up those chemical reactions so that they can happen in real time, so that they can happen quickly and we're not spending days and weeks and months digesting our food, or we would never get energy from it. So a lot of times you'll see graphs similar to this. The red one is the graph without the enzyme, okay? So you notice that it takes a lot more energy and a lot more time. A lot more energy and a lot more time without the enzyme. You look at the enzyme, that curve is so much shorter. A lot less energy and a lot less time. So enzymes speed up chemical reactions. They make them happen much faster than they would without the use of enzymes. Right, moving on to number 3. Enzymes are also specific. So it kind of goes back to the protein and having a really certain specific shape. Shape fits with a specific substrate. So, like a lock in a key, an enzyme has a substrate that it's going to fit perfectly with, that it's going to match to. And it's only going to fit to one substrate. For example, if you think about people who are lactose intolerant, lactase is an enzyme. So if you're lactose intolerant, it means you don't have lactase, so you can't break down lactose. Lactase is the enzyme for lactose, so they have to fit perfectly together. Lactase is not going to be able to break down glucose or sucrose or fructose or any other sugar. It only breaks down lactose. And that's just one example. And later in class, it'll make a little bit more sense. Fourth characteristic about enzymes. Enzymes are reusable. Okay, as long as their shape isn't changed, as long as nothing happens that's a bad thing to the enzyme, it can be reused. After a chemical reaction, it can be used again and again and again. So we don't always need to create new enzymes. They can be used again. They can be recycled. They can be reused. Okay, so we're going to move on and talk about what it's called when an enzyme loses its shape and how it can lose its shape. So if it loses its shape, it's no longer going to have this property. It's no longer going to be reusable, and it's really not going to have any of these properties because it's not going to be able to do its job and fit to its substrate. So moving on, enzymes can be what's called denatured. Think about nature, it's kind of like natural. Denatured means unnatural. It's when an enzyme loses its shape and can no longer function. Okay, so an enzyme does not work anymore because its shape has been changed. This can happen due to changes in temperature or pH. Every enzyme has an optimum where they function best at. Just like we as humans have a body temperature where we function best, where our enzymes and everything else in our body focuses best, which is about 98.6, enzymes are the same way. So you see here, temperature is on this part of the graph, and then you have the rate of the reaction. So you see right here about 10 degrees Celsius, not much going on. Same thing at about 55. But if you look right at about 38 degrees Celsius, there's a whole lot of reaction going on there. The rate is really high. So the optimum temperature for this particular enzyme would be 38. So if it's not 38, if it's like 10 degrees or 55 degrees or even 20 degrees, it's not going to work as well. It's because its shape is going to be changed. So this is due to a change in temperature. It can also be pH. So if something's used to being acidic, if it becomes basic, that's not good either. And you will have to be able to look at graphs like these and interpret them and be able to find out what the optimum pH or temperature is. Okay. So moving on to looking at exactly what enzymes and substrates can look like and how they fit together and what all these things are called. So you have this picture. A is your substrate. That's what the enzyme works on. You need to make sure that you at least label these things. If you want to write the additional information next to it, you can. If you think you can remember, it's up to you. These are your notes. But you need to at least label them, okay? And this is our enzyme. Where they come together is called the active site. It's where the enzyme and the substrate fit together. B, C, and D are all of your enzyme. You can always tell if something is the enzyme in the picture or a substrate in the picture based on its shape. If you look here, B, C, and D are all the exact same shape. That's your enzyme. It does not change. Up here, your substrate comes in, binds to the enzyme, which is called the enzyme substrate complex, and then it splits apart. It's broken down, and that's your product. Okay? Usually the substrate will be on top, the enzyme will be on the bottom, but sometimes they'll flip it and be confusing. So don't focus on if it's on top or bottom, but what is changing versus what is not changing. So your substrate comes in at the active site, binds to your enzyme. You have your enzyme substrate complex, and then that substrate is going to be broken apart. Okay? So for example, if we're going back to digestion with lactase and lactose, it's breaking down that lactose into smaller molecules so that it can be easily digested and used by your body. So we really, really need enzymes to help speed things up, make them happen in real time. But if their shape changes, that's not good. So thank you for watching this video on enzymes, and hopefully you learned something new.